that bacteria can contain, or most all bacteria, can contain an additional loop of DNA. And we're going to call that, or that's called a plasmid. So I've got my bacteria cell, and it's got its chromosome here, but it also, let me go back to my drawing. I've got my bacteria cell here, and it's got its circular haploid chromosome, contains a DNA, it's made up of DNA. In addition to that, it may or may not have a smaller loop of DNA. This DNA is called a plasmid. Now, the deal with these plasmids is it doesn't contain any of the essential genes. All the essential genes necessary for survival are in the main chromosome. That's where that's contained. But these can contain genes, which means a gene is something that can be transcribed and translated into a protein, and that protein can do something, which means this plasmid can give this bacteria cell some extra characteristics. Sometimes I like to think of them as superpowers, something it can do because this plasmid is contained within that gene. It can make a new protein and do something different from that. So these little extra loops of DNA are not required, they're optional, and they're not essential for survival. They can give some really cool superpowers to that. So these are called plasmids. All right, so what types of plasmids do we have? Some of these plasmids, because they're cool, can give them an advantage over cells that don't have these plasmids. All right, so types of plasmids. We have fertility factors, resistance, bacteriocin, and virulence. Let's talk about fertility factor plasmids, and this one's really cool. So if you have a fertility factor plasmid, and I drew this one in black, we're going to call this an F positive cell. So it's fertility factor positive. It has a plasmid that has some extra genes, gives it some extra characteristics, and it's going to infect, and we call that a fertility factor. All right, so what do these genes do? The genes allow this bacteria cell to make a pili. Do you remember what a pilus is, or pili? If there is another bacteria cell in the environment, the genes get transcribed and translated into proteins, and those proteins allow a pilus to form between these two cells. And if you remember what a pilus is, we talked about that in chapter 3. It's a specialized form of fimbriae that allows for genetic transfer. And that's really all we kind of talked about. But the genetic transfer it allows is not the chromosomes. The chromosomes stay here, but sharing DNA with its neighbors, what it's sharing are these little plasmids, these little extra loops of DNA. So what happens is the plasmid replicates. So it replicates itself. So I've got two identical copies, and this pili has formed based on the proteins it made. And one of these plasmids can go into the other cell, and now it has a fertility factor. Now, this only occurs between F positive cells and F negative cells. So at the beginning, when I started this cell had its chromosome, but it did not have any fertility factor plasmids. We call that an F negative cell. An F positive cell is capable of forming this pili between its cell and an F negative cell. Once that happens, the plasmid that's been duplicated can cross over. Once that happens, my F negative cell has now become an F positive cell, and then now he has the ability to do that to a neighbor here. The conjugation of the pilus goes away, and then now he has the ability to do that with his neighboring cells. This is called horizontal gene transfer. We're able to give genes away horizontally. Vertical gene transfer is from mother to child, or from a cell to its progeny. That's vertical gene transfer. Horizontal is giving genes to a neighbor. This is a type of horizontal gene transfer. All right, so fertility factors contains the superpowers that allows it to pilus, which allows it to copy and then share that fertility factor. But there are other fertility factors. There's another one called a resistance plasmid. So let's go back to my original cell. And my original cell not only has an F positive fertility factor, but does that give you enough contrast? I used blue. Let's pick red. But it also has the ability to have a different plasmid, so a different loop of DNA. This one has some different genes on this one. This this one has some genes that make it resistant to a certain drug. So the resistance, this is called a resistance factor. All right, and the proteins that it can make somehow make this bacteria antibiotic resistant. You know the superbugs we're talking about where they have, where bacteria become resistant to an antibiotic? This is how. It's usually not in the chromosome, it's usually these little plasmids that have that. So say I've got an F positive cell, came up to an F negative cell, it forms that pilus, it duplicates the fertility factor and allows that to get transferred into the next cell. Well, what also gets transferred are these resistance ones. Any plasmids will replicate and then also be able to transfer into that cell. This bacteria cell just traded or gave the ability of this cell to be resistant to the same drug this one is. This is how we get those superbugs. Hospitals, you have a whole bunch of different types of things. This can even happen within different species. So you get one bacteria that's causing one disease and it's resistant to this drug, and you get it in a hospital where lots of these bacteria are coming in contact with each other, and now I have different bacteria that are now resistant to that drug. So this is a big concept and one that's really going on. So we have resistance factors. The other types of factors involved, bacteriocin. These factors allow the plasmid that it transfers makes proteins that allows one bacteria cell to kill other cells. So it's bacteriocin, suicide, death. Bacteriocin factors are superpowers, characteristics that allow one bacteria cell to kill another. Why would they want to do that? Well, remember they're competing for nutrients, so that's a survival advantage. Virulence plasmids. We've started talking about virulence factors, virulence plasmids. What characteristics allow a bacteria cell to get in to a human and stay in and avoid detection by the immune system? These are more likely to cause a disease. This also can be contained on a plasmid. So these bacteria can share these virulence plasmids as well. Okay, so that's it for that section. That was a lot. Here's some pictures. This is another bacteria cell where the the DNA has been unpackaged. You can see that, and you can see the individual circle of those plasmids right